Good afternoon, Iman. Okay, so I'm Rahul Saha, and today we'll be finishing up this worksheet. Okay, so the worksheet pertaining to the chapter Principles and Inheritance, right? So we had completed till I think question number 62 and all. So we'll begin from question number 63. Okay, or where it? Wait, I'm just finding out. Yeah, sorry, question number 67. Come to question number 67 here. So it talks about the disease known as Cry-do-cat syndrome. Okay. So Cry-do-cat syndrome is a very, very peculiar disease. Okay. So Cry-do-cat syndrome is specifically that occurs due to the chromosomal deletion. Okay. It occurs due to chromosomal deletion. So what happens is it's a very, very rare genetic condition which is caused by the deletion. Okay. Deletion means a part of the chromosome will be missing from the genetic material on the P arm, the petite arm, or the small arm of chromosome number 5. So you can say option number C, loss of half of the short arm of chromosome number 5. Good afternoon, Vanshika. So we are doing question number 67. So that is the cry-to-cat syndrome. So the children, when they cry, it seems like a cat crying. So it is known as cry-to-cat syndrome. So chromosome number 5. Okay. Chromosome 5 P arm deletion. Okay. That causes cry-to-cat syndrome. Then next question will come to sickle cell anemia has not been eliminated, sorry, from the African population. Question number 68, because what it could be. So just think about it. It's a little hard question, but if you know that heterogamity, okay, or heterozygous condition, not heterogamity, heterozygous condition can lead to, you can say, protective effect, then we will be able to solve this question. In genetics, I had taught you this specific concept. So coming on to question number 68. So it is not a fatal disease, it is controlled by recessive genes, it is controlled by dominant genes, it provides immunity against malaria. So that is the reason it provides immunity against malaria. So the thing is, what you have here is a normal male, or sorry, a normal person would be having hemoglobin adult, both the genes. Adult means alpha 2, beta 2. Okay. This one means fetal. Okay. This one means, you can say Gover 1. This one is Gover 2. These two are the embryonic. Okay. This is the fetal. This is the adult hemoglobin. So HbA is the adult one. If you are completely down with sickle cell disease, sickle cells, you can say anemia, you will be having HbS. Okay. Sixth position will be of the beta chain will be replaced from glutamic acid, or will be replaced by valine, okay, which was earlier glutamate or glutamic acid. But you can have this phenotype also, this genotype also, HbA, HbS. What will it do is 50% of the time your hemoglobin will be perfectly fine, so you won't be showing real effects. Sometimes you may show or display fatigue, but no real effect as such, phenotypic effect. But when the hemoglobin are sickle shaped, 50% of the transcription time or the transcriptional event, so sickle cell anemia, giving us sickle cell erythrocytes or RBC, when the Plasmodium vivax here or Falciparum malei oval, whatever the strain you talk about, it enters, then it cannot really replicate there, right? Because of constrainment, because of the specific valine residue. Okay, so it cannot really degrade the hemoglobin molecule, and okay, and then it will not be able to produce the malaria toxin, right? So we know that if at all the, you can say, toxin produced after the RBC lysis, so after the lysis of RBC, there is a specific toxin produced known as hemozoin. If this can be prevented, malaria can be prevented. So this is produced in a very less or no amount, okay, because the sickle cell RBC do not support the production of or the proliferation of plasmodium protozoan. So it provides a specific, you can say, protective effect to those populations. So it's an evolutionarily derived concept. Okay, evolutionary derived concept. Now next is 69, pretty easy question. Okay, same question. Okay, you can see here round seed, capital R, capital R, capital Y dominant, that we have already seen. Okay, so these are the questions which are pretty easy, but they require specific attention only. Otherwise, these have nothing to do with the, you can say, conceptual prowess, because it's a very basic question. So the answer would be here: round seeds with yellow cotyledons and wrinkled seeds with yellow cotyledons. So what are the expected phenotypes in F1 generation of RY? Okay, so the thing is, here yellow cotyledon was dominant over green. Okay, so round and yellow. Okay, and you can say expected phenotype RRYY. So what you will get is, straight away you will get wrinkled seed, and also since yellow is dominant over the green, okay, so what you will get is option number A would be the correct option. So how many different kinds of gametes, come to question number 70, how many different kinds of gametes will be produced, okay, by a plant having the phenotype? So this needs a little bit of trick. Okay, very easy. So how many gametes, okay, can be produced from, you can say, plant with capital A, capital B, sorry, capital B small b, and capital B small c, sorry, capital C, like, so how many gametes can be produced? So 2, 4, you can say 3, 9. Question number 70. So how will you be able to solve it? Okay, the answer is 2, but how did you get the answer? Okay, so the thing is, the plant having genotype capital A, the way to solve this is, okay, don't go by the answer and try to memorize it. There is a special technique to solve this question. Okay, you can see this, the plant having genotype capital A, capital B, small b, capital C, has how many heterozygous characters or alleles? Only one heterozygous allele. Okay, so number of gametes, okay, will be 2, because you put the formula 2 to the power n. N is the number of heterozygosity. So n is what here? N's value is 1. Only 1 is heterozygous here, not 2, not 3. So 2 to the power 1 will be 2. Okay. So those two gametes are this one, A, B, C. You can check theoretically also. Then capital A, small b, capital C, because this and this are same. So you can only change this one. So only two gametes will be possible. This is the theoretical explanation. This is simply the shortcut, the formula for that. 2 to the power n, n is the number. So here the maximum number of heterozygous could be 3. If it is 3, 2 to the power 3 means 2 into 22. Okay, so 2 into 2 will be 4, 4 into 2 will be 8. So maximum 8 gametes could have been produced if all were heterozygous like this. Okay, so this is the way this question is solved. Now question number 71, again an easy question. Okay, so today we'll finish it off. Okay, this worksheet. So phenotype of an organism is the result of 71. Pretty easy. Genotype and environmental interactions. Okay, because environmental interactions, they do play a great role, because we talk about evolution, and especially when we talk about epigenetics, the histone part that I taught you. So sometimes what happens is, genotype is there, but phenotypic manifestation, sorry, environmentally induced manifestation does not occur. Like suppose you are susceptible to multiple sclerosis, which is an autoimmune disease which will degrade the myelin sheath. Okay, that's the layman term for multiple sclerosis. So when myelin sheath is attacked by our own immune system, autoantibodies, what would happen? Conduction will be reduced, you will be having, you can say, nerve damage, nerve tingling, burning, you can say lesser control, eye problems, vision problems, all that will be happening. Now, it has been seen that if a person susceptible to, if he has a genotype susceptible to multiple sclerosis, that is MS, an autoimmune disorder, because here your body's immune cells are attacking your own self antigen. It is known as autoimmune disorders. Okay, you might have read it in chapter 8. So here, if that person takes vitamin D3, cholecalciferol, okay, at least 2000 international units, okay, it's known as 2000 IU daily, okay, so then that person would remain protected for his entire life. He may not develop. Again, it's an environmental thing because vitamin D is causing what? Epigenetic silencing, okay, of that specific protein which is susceptible. Okay, it is not letting it express too much. That expression, de-expression via epigenetic mechanism, histone acetylation will lead to more expression. Histone triple methylation will lead to lesser expression, depending on the residues again. That is not in your course. So, which triple methylation at which lysine residue can also lead to overexpression. The thing is, you play with epigenetics there. Okay. So, coming to the next question. Which one of the following is an example of polygenic inheritance? Again, it's a very easy question. That is skin color in humans. Very easy question. Okay, multiple genes are required for developing the skin color in humans. Now, in order to find out the types of gametes produced by a pea plant given having a genotype A, B, B, it should be crossed with the genotype. Question number 73. This is talking about a test cross. Okay, which one? How will you find out what is the parental genotype? You cross it with the homozygous recessive one, small b. You cross it with this. Okay, whatever, if it is AA, okay, or if it is BB, if you get 50%, okay, and if you get, you can do two monohybrid crosses again. If you get 50% like this, okay, B, B. So 50% if you get recessive characteristics, that means you are heterozygous for this trait, right? Very simple. So you can see there are many questions pertaining to test cross here, various types of test crosses, okay? Now G6P deficiency, dehydrogenase deficiency, it is a very important question, okay? Glucose 6-phosphate dehydrogenase. Now, first of all, I'll tell you what is dehydrogenase. Okay, if you have studied properly in class 11th biochemistry, okay, you will be knowing dehydrogenases. Okay, so glucose 6 phosphate disease, okay, 6 phosphate dehydrogenase, okay, so that is a cytosolic enzyme which catalyzes the reaction of, I'll tell you the reaction. There is a specific pathway, okay, known as hexose monophosphate shunt pathway. So what it does is it catalyzes that specific reaction. I'll first write down the reaction for you, then I'll explain this question also. So glucose 6 phosphate, okay. This phosphorylation 6 phosphate you can get from the glycolytic pathway also, okay. So the thing is, glucose 6 phosphate when you have got, still it hasn't entered glycolysis. Don't think it has entered unless and until it is converted to fructose 6 phosphate isomerase. Then you have an irreversible reaction PFK1, okay, where ADP, sorry, PFK1 and coenzyme this one, here ATP will be broken down into ADP plus PI. Phosphorylation at the first position will happen. It will become fructose 1,6 bisphosphate. Okay. When it becomes fructose 1,6 bisphosphate at first position of carbon, sixth position of carbon, then only this that glucose that is started from here, okay, became 6-phosphate by hexokinase. Then only this glucose is destined for glycolysis. So this step is known as rate limit. You can say first committed step, fructose to fructose 1,6 bisphosphate. First committed step towards glycolysis. And proper NEET question this is. Okay. Like when will the body decide or the cell decide that now only pyruvate will be formed? Now it will not diverge into any other pathway. Okay, because there are multiple pathways concerning glucose, not only glycolysis. Okay. One of those pathways is hexose monophosphate shunt pathway. So if you are at this stage glucose 6 phosphate, it may go down this route or it may take another pathway. So if it takes another pathway means if the products too much pyruvate is there and also or too much fructose 6 phosphate is there, so too much you can say glycolytic end products are there, then it will diverge into hexose monophosphate shunt pathway. Okay, it will use the enzyme glucose 6 phosphate dehydrogenase and it will produce 6 phosphogluconate. Okay, and dehydrogenase means these are the enzymes that take part in oxidation reduction reactions. Okay, so this is an oxidation reaction. Okay, so here something must be reduced. So NADP plus is reduced to NADPH plus H plus. So oxidation reduction, it goes side by side. So this is an oxidation reaction. So this is a reduction reaction. Okay, always know the basics of biochemistry. Okay. So without reduction, if I just do oxidation means it has lost electrons, that those electrons can go and bind to any other significant molecule in the cell like DNA, like protein, like lipids and can even cause cell death later on. That is known as free radical damage. It can produce free radicals like superoxide radicals. Okay, this ones. Then it can cause peroxide, okay, hydroxyl radicals. These are very, very lethal, you can say highly reactive radicals that react with multiple components of the cell and will damage it. So you need to quench that electron. So who is the quencher? NADP plus here. Okay, nicotinamide adenine dinucleotide phosphate. It becomes nicotinamide adenine dinucleotide phosphate hydrogen. So this is the reaction. And dehydrogenases, they catalyze oxidation reduction reactions. So they fall under the category of oxidoreductase in the enzyme category. We have six types of enzymes, right? Oxidoreductases are there, lyases are there, ligases are there, right? Then transferases are there. You might be knowing. So this falls under the oxidoreductase category. Now, they say that glucose 6 phosphate dehydrogenase deficiency. Okay, what is that deficiency and what happens in that deficiency? Okay, so glucose 6 phosphate deficiency is an inherited condition. We know that because the gene will be mutated. Okay, so what happens is in this deficiency, okay, it affects mostly the red blood cells. Okay, mostly it affects our red blood cells. Means the erythrocytes. Always know this. D6PD deficiency always, okay, you can call you can say it affects the RBC. Always remember. Now, since 6-phosphogluconate cannot be produced, okay, also this is called as the pentose phosphate pathway. Now, the you can say what it is doing. You can say it is regenerating here. Just look here. It is regenerating NADPH. Okay. So NADPH when it is regenerated, what it does is, okay, when NADPH is regenerated, NADPH will do what? It will regenerate. This is known as oxidized glutathione. Okay, This is known as reduced glutathione, GSH. Since this is an reduction reaction, now this NADPH is needed. The NADPH will be converted to plus H plus NADP plus. Now this reaction will continue. So you can see this cycle will keep on going. NADP plus produced. Now it is again becoming NADPH here. Okay. Now again this one. Now I I'll tell you that glutathione. Okay. Glutathione, and the enzyme used here will be glutathione reductase. Okay. So glutathione reductase. Now glutathione is a very important, the most pivotal, you can say, antioxidant which will quench all the radicals like this. Okay. Now when it quenches, it becomes oxidized to GSSG, oxidized glutathione. Now that is of no use. It has to be again reduced to reduced form of glutathione, GSH. When reduced, it must oxidize something. Now its reduction, the oxidized glutathione cannot be converted to the reduced glutathione without the presence of NADPH. And NADPH is generated from this pathway, and NADPH cannot be generated without this enzyme. So too many oxidant radicals will be accumulated in the red blood cells. Okay, and lack of G6PD then will lead to hemolysis during any kind of oxidative stress. Okay, that is, again, that can occur due to medication, or one good question in NEET is asked that fava beans, if you take, you will die quickly if you do not, if you have G6PD deficiency. Okay, that is the entire cycle behind this question. Okay, so it is associated with hemolysis of, you can say, RBCs. That is why, that is the explanation of this question. Okay, so can come to question number 75. Okay, so a woman, okay, a man and a woman only. So that is generalized question. Okay, a man and a woman. Okay, and a woman who do not show any apparent sign, who do not show any apparent sign of a certain inherited disease. Okay, a man and a woman who do not show any apparent sign of a certain inherited disease have 7 children. Okay, 2 daughters and 5 sons. Okay, 3 of the sons suffer from the given disease, but none of the daughters affected are affected. Okay, which of the following mode of inheritance do you suggest for this disease? Okay, so the thing is, parents are not showing any signs of inherited disease, but sons do show. Okay, hence it cannot be autosomal. Okay, or sex-linked dominant disease, as dominant mode of inheritance is seen in all the generations that is there. Now the mode of inheritance of such given disease must be sex-linked recessive, and the defective gene is present on the X chromosome. That is how you deduce it. So in the given case, what is happening? The father is having normal genotype, capital X, capital Y. Okay. Man is having capital X, capital Y. Woman is having one defective. She is the carrier, you can say. XC, she is the carrier. Now all daughters will receive at least one normal copy of X chromosome from father. Hence none of them will show the sign, and only the sons will show the signs. So what will happen? 3 out of 5 sons, they received the defective X chromosome from the mother and suffered from this disease. Okay, so that is a very easy question again that you need to know. So it must be sex-linked recessive. Okay, 3 copies of chromosome 21 is considered as Down syndrome. Question number 76 is Down syndrome. Okay, then hemophilia, very easy question. Okay, 77. So again, it is due to an X-linked recessive mutation. Okay, these are all repeated questions. I'm going quickly. Now, which of the following is not a hereditary disease? Which one? You can think. Is not an hereditary disease. Okay. So, answer is here. Thalassemia is a hereditary disease. Hemophilia is a hereditary disease. Okay. So, when we come to cretinism, so cretinism is an inherited disease. So, cretinism means you can say is not a hereditary disease. Okay, that is sorry. Cystic fibrosis thalassemia. Okay, that is also hereditary. Okay, here I don't think anyone is the option because cretinism also is a hereditary disease as far as my knowledge is concerned because it is congenital hypothyroidism. Okay, this sometimes this becomes a you can say I think there is some mistake here. So we'll switch on to question number 79. Okay, so these kinds of questions we had done already with normal vision. Okay, so we can skip this one. We can try it. If you can't solve, then you can ask me later on. Now question number 80. So we had done multiple questions like this. Okay, so in a plant, red fluid dominant are dominant over shortness. Okay, so this question also you can try on your own. Very easy question. Okay, question number 81. A male human is heterozygous for autosomal gene A and B and is hemizygous for hemophilus gene H. Okay, so this is a good question. Let's try this one. Question number 81. A male human is heterozygous for autosomal genes A and B, okay, and is hemizygous for hemophilic gene H. Okay, now how to solve this question? The thing is, just to make you perplexed, this question has been given. It's quite easy. Okay, so when a human male is heterozygous for autosomal gene A and B, and also he is hemizygous for hemophilic gene H, then his genotype will be what? Genotype will be, you can say AA, BB, okay, and capital, you can say X. I can put XH, okay, and Y. This is his genotype, capital XH and Y. As why did I put XH? Because hemophilia is a sex-linked disorder. Now the gametes produced, okay, from this genotype will be what? He can produce multiple genotypes A, B, XH, then A, B, Y, then A, B, capital XH, okay, that is capital only, okay. Then he can produce A, B, Y, okay. Then he can produce A, then B, then capital XH, then A small by, you can produce A capital B XH, okay. Then he can put AB, or he can produce one more gamete A capital B Y, okay. So the total number of sperm that carries ABH, okay, is how much? ABH. So H here means XH, right? Is only, you can say, how many are there? Small a, small b, okay, small x, xh. Xh means H, right? So this one is only 1 out of 8, right? Is only 1 out of 8. Small a, small b, small this thing. So that is 1 upon 8, that is 12.5%. 1, 2, 3, 4, 5, 6, 7, 8. So it is 1 upon 8, small a, small b, and H means XH. We should look for this ABH phenotype from the, means this one, XH, which I am calling it, from the genotype. So it becomes 12.5%. Okay, 1 by 8. That is correct, 1 by 8 only. So, question number 82. Lack of independent assortment of two genes A and B, okay, will lead to, or is due to, you can say it's due to simply linkage. If linkage is there, again there is a specific question. If linkage is there, what will happen to Mendelian inherit, or which law of Mendel would have been, you can say, ablated or affected adversely? It is again law of independent assortment. Okay. So question number 83 you can try on your own. Okay. 84 also you have tried hundreds of questions, like many questions like this. 84 you can try. Now question number 83, 84 you can try. If you have any questions you can ask me on my number. Now 85, if we check, the recessive genes on X chromosome in humans are always, okay, expressed in males. Very easy question, this one also. Okay. Okay, what's the recessive feature? This is pretty easy. 86. Okay, that is the green seed color. Now 87, the genes controlling the 7 characteristic are now known to be located on how many different chromosomes? Okay, so they are now known to be located on, you can say 87, you can say 4 different chromosomes. Okay, that's a new thing for you. They are different, 4 different chromosomes these genes are located on. Okay, now 88, reversed in one cross, okay, is known as reciprocal cross. Okay, it reversed means it is known as reciprocal cross. Now 89, pattern baldness. Okay, that is a good question. Question number 89. So when we talk about pattern baldness, that is mustaches and beard in human males are examples of, that is known as sex influenced traits. Okay, that is known as sex influenced traits. So which of the following conditions, through though harmful in itself, is potential savior from a mosquito borne disease? Okay, again sickle cell anemia, that same heterozygosity thing, sickle cell anemia. Extra copy comes on 21. What percentage of offspring produced by an affected mother and a normal father? Okay, question number 91. Affected mother and normal father. So we know that it could be what? It could be almost, you can say, 91 for 91st question. Affected mother is there. So she will produce 2 ovum. Okay, 2 ova. Okay, suppose. So okay, one of the, you can say, from oogenesis you have this one. Okay, so what you will be having is production of ovum. So one ovum is produced, okay, per cycle. So the thing is, it if the mother is affected, okay, so if the mother is affected, then the dynamics changes. Okay, Down syndrome dynamics changes. How does it change? So Down syndrome, we know that it is caused by an extra copy of chromosome number 21. Now I told you that non-disjunction theory is there. Okay, so non-disjunction theory, when we talk about, okay, what does it mean? So that means that it chromosome number 21 at the diplotin stage, okay, at the diplotin stage, what will happen is the resolution of the holiday junction, or the, you can say, this one, the RUV B complex, the REC BCD, they are not properly, they have not been able to dissociate. So disjunction has happened. So how to solve this question? Now we know that Down syndrome is an autosomal recessive disease. Okay, not recessive, sorry, autosomal aneuploidic disease. Okay, Now, according to the question, affected mother carries 47 chromosomes, means 45 plus XX, not 44, okay? And normal father has 44 plus XY, okay? Now let's make the gametes. So we'll talk about gametes. 45 plus XX, it will make, you can say, 23 plus X, okay, that could be the case. And this will make 22 plus X, okay, that will be done by, you can say, 44 plus XY, okay. Now 22 plus X is, sometimes you can also make, you can say, 22 plus Y, 22 plus X, that is both the cases, sorry, this one I can make it like this only. One of the gametes will be 23 plus X, sometimes the gamete will be 22 plus X also in this case. In case of father, okay, I can write it down here, it will be 22 plus X and 22 plus Y. Now, in this case, what will happen when it goes with 23 plus X, okay, it will become this, and this I am going for. It will become 45 plus XX, and when this goes with this, it will become, you can say, 44 plus XX. Now this is diseased, this is normal. Same way, next gamete, it will go with 25, 45, you can say, XY, diseased. And it will go with 44 plus XY, that is again normal. So 50% is the, you can say, trisomic, and 50% normal child. Okay, so answer will be your 50%. Okay. Now, Drosophila melanogaster, we have talked about this pretty clearly. Okay, so the ratio of X chromosome to the pair of autosomes, they determine the sex in Drosophila. Okay. Now 93, which of the following is an example of pleiotropy? Okay, so we can consider again 93rd question to be, you can say, C, right? Pleiotropy, polygenic inheritance and pleiotropy, multiple effects. So one can be having malarial protection effect, that another is the sickle cell normal effect, right? Sickle cell anemia. Now question number 94, there are three genes. Okay. Now this question will be good. Now there are three genes. There are three genes A, B, and C. Okay. A, B, and C. Now percentage of crossing over between A and B is 20%, B and C is 28%, and A and C is 8%. Okay. Now how to find the sequence of genes in the chromosome? Now this is known as chromosomal mapping. Okay, how to find? Now just understand, I'll tell you in short, there is a long process also. Okay, I'll tell you in short one how to do chromosomal mapping. Good afternoon, Tarun. Good afternoon, Ayushi. So I'll do the, you can say, shorter way. Okay, so the thing is, the shortcut method here is following A, B, and C. So since recombination frequency is directly proportional to the distance between the genes, okay, if more genes, more recombination will occur. Okay, one more thing I'll tell you, recombination, you can note it down. Frequency means events pertaining to recombination is directly proportional, okay, to the distance between the genes. to the distance between the genes, right? Now, we can say the values are used to locate the genes on the chromosome directly. I'll tell you how it is done. Now, here there are three genes, okay, A, B, and C. So, you can assume any sequence. Let's assume any sequence. You can assume A first, B first, then C first, or A, C, B, or you can assume B, A, C, okay, which depends on the gene present in the middle. Whatever genes will be present in the middle, it will become very, very easy for us to deduce, okay. So the combination frequency between A and B, okay, A and B is 20%. So A and B. So let's make this combination frequency as X. X is 20%. Then A and C, we can call it Y. That is 8%. Then B and C, I can call it Z. That is 28%. Now, as we can see that, you can say Z is equal to X plus Y or not. Just check. Z is equal to X plus Y. Here, Z is 28%, X is 20%, Y is 8%. So Z is equal to X and Y, X plus Y. Okay, this is a shortcut method that I am telling. This means that genes B and C are present at extremes, okay, and you can say A is in the middle, okay, A is in the middle always. So A will be in the middle, B and C is in the extreme, okay. So it will become BAC. This is a shortcut method. This is the shortcut method of deducing, okay, the entire gene mapping. Okay, long cut method I'll be explaining to you, okay, in the next few classes. After this chapter is done, we will be explaining the long cut method and doing also association questions. Okay, genetics chapter, it has molecular basis. So many questions in NEET are there which are not at all discussed even in NCRT. Okay, so I'll discuss the longer method also, which will give you more of certainty towards solving these questions. But keep in mind, this shortcut method is very, very quick method. This one, okay, you can solve it quickly. Now coming to the next question, because PMT doesn't require you to know the long cut. Okay, shortcuts are very, very essential. So I told you shortcut also here. So on selfing of F1, genotype AAB, genotype F2 generation. Okay, so that is very, very easy. Okay, question number 95. This is the trihybrid crossing. Okay, so again only two gametes will be produced. Okay, answer will be A only here. A gene is said to be dominant if, very easy question. Okay, question number 96. Okay, means it has to express in both heterozygous and homozygous condition. Okay, then question number 97. A diseased man where is a normal woman, they get 5 sons. Okay, and all sons were normal. Question number 97. 3 daughters and 5 sons. All the daughters were diseased and sons were normal. Okay, so what does that mean? Marries a normal woman. Okay, diseased man marries a normal woman. All the daughters were diseased, sons were normal. That means what? It is sex linked dominant or not? Like he had this type of disease. Okay, XDY. He marries a normal woman. Okay, so since this one, the sons are normal because they are getting X from the one, they are normal. But even if they are getting this one, if they are getting proper chromosome also, suppose one is carrier, let's suppose one is carrier, but suppose this one is proper. So since this one is proper one, so even if it is getting this one, okay, proper one, still the female is having disease. That means what? It is X-linked dominant disease. Okay. So 98th question. Which of the following is the correct match? Okay. Down syndrome, 121st chromosome, very easy. Okay, two non-allelic genes produce the new phenotype when present together but fail to do so independently. Okay, again an easy question. Fails to do so independently. That is known as epistatic. Epistatic gene and hypostatic gene, which we have talked about. That is dominant epistasis, recessive epistasis, and all. So A and B genes are linked. What should be the genotype of progeny in a cross between AABB and ABAB? Okay, A and B are linked always. Okay, question number 100. So they will never be dissociated. Okay, so it will be A and B are linked. So A and B will stay together. So it could be capital A, capital B, small b, small a, small b, small b. So A and B you cannot, you can say, remove. Okay, from one, you cannot separate, you can say. So the answer will be option number B. When dominant and recessive alleles express together, okay, very easy question. Codominance, ratio of complementary genes is, okay, that is 9:7. I'll explain it to you how. Okay, complementary genes, ratio of complementary genes. Again, you can see this is not in your NCERT book at all, but it has been given in your, you can say, question. So the complementary genes are what? The complementary genes are two pairs of non-allelic, you can say non-allelic dominant genes present on separate loci. Separate loci. Okay. Now they interact to produce, they interact to produce the phenotypic trait. Okay. Produce, you can say, only one phenotypic trait. But neither of them, okay, next line is very very important, but neither of them, if present alone, if present alone, produces the phenotypic trait. In the absence of the other. You have seen this complementation. I have taught not the ratio, but the one thing I taught you, that the blue white skinning, if you remember, XGAL 1. So we know that there was this thing known as LAC Z delta M15 mutant. Okay, beta galactosidase containing LAC on the E. coli, you can say, circular nucleoid DNA, means genomic DNA, and the plasmid contains another, you can say, truncated part of the beta galactosidase, which only codes for 146 amino acids. Okay, so when this gets transformed, it will have an amp R here. If it gets transformed with the non-recombinant one, this LAP 146 will complement this rest of the beta galactosidase gene, Okay, and the lack of opamine will be proper, the beta galactosidase gene will be proper, and then IPTG will act as the inducer. Okay, so after that, what you will see is you will see the breakdown of XGAL. Okay, so XGAL will be broken down into, you can say, 5 bromo, okay, 4 chloro, indoxyl, and after that you can say it dimerizes. Okay, if you remember that, it dimerizes to form, you can say 5 prime bromo, 4 prime chloro, okay, indigo. That is what happens. Okay, so after that what you will see is you will see the blue color. So this is known as alpha complementation. This one, when it is present with this one, then the truncated, both the truncated parts are present together, and then we are able to see completion of the beta galactosidase gene and beta galactosidase, you can say, protein or enzyme working, breaking down the XGAL. Okay, breaking down the XGAL to produce this one, which will dimerize and form the blue color. And once this, at this position is the position of the MCS also, it overlaps. So if the insert is here, this gene is insertionally inactivated. Now if you have recombinant plus, you can say, transformed, now what will happen is this is not going for transformation, sorry, it is not going for complementation, and then there will be only white colonies. That is why ampicillin is put here, so that it first grows in ampicillin. Otherwise, white colonies could be anything. Nothing grows in, still it is white. That is why we put ampicillin. So this is an example of alpha complementation as well. Okay. So when you go for complementation analysis, okay, so the ratio that we get is 9 is to 7. Okay, it changes to 9 is to 7 from 9,3,3,1. Okay, so that is the meaning of complementation. Okay, so once you go for complementation, there is, you can say, different ratios are there. So it will be 9 is to 7. You can just memorize this ratio. It will change from 9, 3, 3, 1. Okay, in the, you can say, practice classes that we'll be conducting very soon, I'll be describing this in detail. Okay, how this comes 9 to 7, if you want to know, because it's not at all given in your NCRT. If you want to know, I'll, you can say, explain it in detail completely. Okay, how it is 9 to 7 only, in why not 9,3,3,1, how does the ratio change, on what aspects does the ratio change. Okay, so those things will be explained in detail. Now the next question, you can have is independent assortment of genes, right? That is the next question. Independent assortment of genes, question number 103, is not present. Okay, is or does not take place. Okay, does not take place when, question number 103, when genes are linked and located on the same chromosome. Very easy question. Okay, then sickle cell anemia is induced due to, very easy, again you can do it yourself, change in amino acid beta chain. Okay, number of bar bodies in XX female, okay, 105, okay, is 3. 3 why? Because only one chromosome remains activated, others are inactivated. I had told you this formula, this thing, that X inactivation, why? Epigenetics, so that the number of X proteins in male is equivalent to female, but otherwise females, due to transcription, will produce double the amount of, you can say, proteins, which are, with genes, double the amount of genes will be expressed which are present on the X. So that will make profound physiological difference between a male and female. Okay, because X contains H2 and many other, not only sex genes. Okay, not only the genes carrying or responsible for secondary sexual characteristics. So if you have four X's here, so only one needs to be activated, deactivate the three, and each deactivation results in the formation of bare body. Okay, so you have complete three bare bodies. Okay, the entire, you can say, mechanism of bare body formation, okay, epigenetic bare body formation mechanism, is not at all discussed in your course, but still you can see, okay, questions from bare body are still, you can say, being asked in NEET examination. That is why I say in biology you have to be very very prudent while going for NEET. It's not always NCERT at all. Okay, so only 60 to 50 to 60% are NCERT based questions. The rest will be, or will try to, you can say, trump you, right, in these questions. So 106th question, okay, that is, male XX and XY sometimes occur due to, you can say, transfer of segments in X and Y chromosome. Very very easy. Okay, now probability question is there of four sons to a couple. 107, probability of four sons, okay, to a couple, that is there. Now how will you find out probability of four sons to a couple? Probability of four sons to a couple is, so let's do it. So because the chance of having a certain sex of child is 1 by 2, right? Very easy. Little bit mathematics, is 1 by 2 certain sex in a child is 1 by 2. Okay, and they're all independent of each other, so the probability is simply, okay, four children, independent events, means law of multiplication that I had explained to you in the genetics part, the shortcut method of dihybrid cross. Okay, you can do two monohybrids and go for simultaneous multiplication because they're independent events happening simultaneously. So 1 by 2 to the power 4, because they want 4 sons to the power 4. What you will get is directly 1 by 16. Very very easy. Just you should know which formula to apply. So here you will apply the multiplication rule because the events are independent and occurring simultaneously. So multiplication rule is applied, just the same rule which I have made you apply, but I taught you how to apply it in the dihybrid cross. You want a shortcut to 9331 without doing the dihybrid cross, without going for the huge cross itself. Okay, so just try out the rest of the questions, okay, from this sheet. Okay, so from, and just tell me on my personal number which questions, because triple X syndrome and all this is all has been taught. Easy questions these are. Just try it out. Okay, we'll again look at some of these questions. Okay, and proceed with the next worksheet in the next, you can say, class. Just give these questions 10 minutes and then proceed with the next worksheet because it's all easy. So if you have any problem, you can ask me during the next week regarding the questions that are remaining in this worksheet. Okay, and then I can specifically discuss them in the next class and proceed with the next worksheet. Or I'll just summarize this worksheet, the difficult questions, and do it in the next class. Okay then. So if you had any problems, you can ask me on my personal number. Okay then.